So it's my great pleasure to introduce our first set of speakers tonight, Damgar and Ben O'Mara, and they're going to talk about the isolation and characterization of novel mycobacteriophages, which is what we do in the phage lab. So I'll hand that over to them.
Hello, everyone.
Hello. Good evening. Hope we're all doing well today.
Sorry, I'm trying to share my screen.
You got this, Tom.
Here we go. All right, all good, hopefully. All right, good evening, everyone. I'm Dominique Gar. I'm here to present to you about the isolation and characterization of Mycobacteriophages on behalf of the Phage Genomics Lab at the University of Colorado Boulder. I'm Dominique.
And hello, my name is Ben O'Mara, and over the course of this past semester, we've been isolating and archiving specific microbacteriophages. So what exactly is a microbacteriophage? Well, they're these cute little green guys that you can see on the bottom right. However, if you're a bacteria, trust me, you're not going to think they're cute, because these microbacteriophages are actually a specific type of virus that target and destroy bacteria. So to better understand these microbacteriophages, which we're going to be calling phages for short and simplicity, we can take a look at their structure as seen in the diagram on the left. So looking at the top of the structure, we can see that there is a caspid head with a nucleic acid core, which is the region in which the genomic information of the phage is stored. As you look further down the diagram, we can see the tail region, which is notably comprised of the tail sheath with the distal tail fibers.
These tail fibers become important because they specifically bind the proteins on the surface of bacterial cell, which is the start of the infection process. Once the phage is bound to the bacterial cell, it injects its genome into the bacterial host. There, it will circularize and begin to use the mechanisms that exist already within the bacterial cell to replicate the phage and its genome. The next generation of phages will lyse from the cell, killing the bacteria and restarting the infection process. This is known as the lytic life cycle and is the only life cycle that lytic phages can undergo.
However, there's a second life cycle that some phages can undergo, known as the lysogenic life cycle. During this life cycle, there's a divergent after the phage has injected its genetic information within the cytoplasm of the bacteria, and then it's going to integrate the phage genome into the bacteria's genome, creating what is known as a prophage. This prophage will then exist until later it recombines in the cytoplasm and can re-enter the lytic phage to complete its life cycle. Phages that can undergo both the lytic and lysogenic life cycles are known as temperate phages. The key to take away from this is that the lytic cycle can kill bacteria, meanwhile the lysogenic cycle only integrates into the genome.
To understand which life cycle a phage is going through in lab, we isolate the bacteriophage and then plate it on an L-agar plate, as you can see here, with some M-smeg bacteria, allowing the phage to infect it, and then we can physically observe the plaques caused by this infection to decide what life cycle the phage is going through.
So looking at the plaque morphology specifically, which morphology just refers to the physical characteristics of it, we can first look at the phage neowise on the left. We can see that on this M-smeg bacterial lawn that there are these plaque sites, which are the regions in which the phage has targeted and destroyed the M-smeg bacteria. Due to the clear and clearly defined nature of these plaques, we can name the phage neowise as a lytic phage because it is only destroying the bacteria during the lytic cycle. As compared to the phage spicy and the phage spatula, we can see a more cloudy morphotype associated with these phages. The cloudiness is a result of some of the phages undergoing the lysogenic cycle, which, as you remember, doesn't actually kill the bacteria. So that means that there's some living bacteria that have prophages in it and some bacteria that are completely destroyed, creating these cloudy plaques. So we can identify the phage spicy and the phage spatula as both temperate phages. So to further our journey along classifying phages, we can take a look at two-tailed microbacteriophage morphotypes, specifically the Syphoviridae and the Myroviridae. On the left, we have the Syphoviridae, which are characterized for having these long, flexible, but non-contractile tails with these short little stubby tail fibers at the distal end. We can compare this to the Myroviridae, which have these complex and contractile tails. However, they are non-flexible. And additionally, they have much longer tail fibers at the end.
To observe these morphotypes in lab, we take an electron micrograph image of the phage in order to see the physical attributes of our phages, which is super exciting. Here we have 2 Sympoviridae isolated by Ben and I ourselves. These are Sympoviridae, much like every phage that was isolated this semester, and you can tell that because they have the smaller head than tail size. Although even between 2 Sympoviridae, you can see that there's a lot of diversity between Spatula and Taz over here. And so you know that there's a lot more to look into even within the same morphotype, which is why we decided to look into phage DNA. So here we have looked into the phage DNA to classify what cluster it's in. And a cluster is how we group phages together based on their sequences that are within their genome. So here you can see 4 different phages and their gene maps. The color between the gene maps signifies homology between sequences. So at the bottom, you can see A7 and B1 cluster phages with no stripes between their genomes, which signifies that there is below 50% similarity between their sequences, which is what places them in separate clusters. However, you can see 2 A cluster phages in the middle, A1 and A7 phages, and they have some stripes of color between their genomes, signifying that they have some homology between their sequences. They are at least 50% similar, which is why they are in the same cluster.
However, we can take cluster classification one step further by looking at the subclusters. For 2 phages to exist within the same subcluster, they have to have a similarity in their genetic sequence of at least 70%. We can see a clear example of this between the 2 A1 phages located towards the top of the diagram. As you can see, there are a lot of these purple regions in between the 2 genomes, again signifying similarity between the 2 sequences. So we can hypothesize that both these A1 cluster phages, which exist within the same subcluster, will have similar functions and structures.
Now, we can't sequence the entire genome of every phage, so what we do instead to look at their genome is run a restriction digest analysis in which we isolate the DNA from the phage and we mix it with several restriction digest enzymes, which you can see labeled at the top of the gel. These enzymes cut up the DNA into little fragments, which we then run through gel electrophoresis to physically observe the fragments of the DNA. So here you can see the bright green band signifying fragmented DNA. We can then count these fragments and cross-reference the fragments in this gel with gels of phages that have previously had more sequencing done, which allows us to kind of compare the genomes of phages from our lab to phages that have already gotten placed in a proper cluster. This allows us to guess the cluster of our phage based off of the cuts in the gel. It's an imperfect process because of how hard it is to count these cuts and due to other imperfections that may show up in the gel. However, it allows us to get a good guess, which we will test further when we decide the actual cluster of our phage next.
So the lab technique that we use within our lab is known as cluster assignment via polymerase chain reaction to further figure out which cluster a phage belongs to. The best way to understand this process is by taking a look at an example from our lab, which was with the phage Fraggle. So during the restriction digest procedure, it was determined that Fraggle was either an A3, A4, or K1 cluster phage. So then we use the PCR to further figure out which of those 3 it belongs to. The way the PCR test works is it will take a cluster phage DNA and add it to a specific primer with the hope that those are going to match and create a positive band. We can see this under the positive control in which we took an A3 cluster phage DNA and combined it with A3 primer, which then resulted in a replicated genomic sequence which appeared on the gel. However, in the negative control, we took another non-A3 cluster DNA and combined it with A3 primer, which resulted in none of that light green band appearing above the primers at the bottom of the gel. So when we look to the right to the phage Fraggle, we can see that there was a positive band on the right underneath the A4 primer, showing that Fraggle is most likely an A4 cluster phage.
Once we understand the fullest extent that we can about our phage, we can apply it to the real world, specifically in phage therapy. For example, we have Isabella here, who suffers from cystic fibrosis, which leaves her immune system weakened and her lungs highly susceptible to bacterial infections. Isabella was previously treated with antibiotics, but her bacteria in her body started to build antibiotic resistance, which is becoming an increasingly intense crisis around the world as we overuse antibiotics. So what they did is they turned to phage therapy. On the right of the screen here, you can see 3 phages isolated by the C phages foundation. And what they did is they found out that the phages were lytic and specific to Mycobacteriophage abscessus, or Mycobacteria abscessus, sorry. And the phages were used in cocktail together and implemented into Isabella's lungs, where they actually were able to successfully kill the bacteria in combination with antibiotics, saving Isabella's life, which is super cool. And we hope that phage cocktails can be further implemented into the medical field to save many more lives in the future.
So the way in which these wonderful success stories tie back to the research that we did in our lab is that in this lab, we were able to create phage samples of these specific phages along with the archive information that we gathered from the various lab techniques we went through. So an example of this is that we're able to send these samples of phages to the University of Pittsburgh, where they can put them in an archiving freezer, such as the one that you can see here. That way in the future, when doctors or scientists want to try and create a phage cocktail for phage therapy, they can then go to archive freezers such as this and find the phages that are right for the job. However, phage therapy goes beyond just the medical world into other applications, including the agricultural field and pathogenic detection. So ultimately, phages are the future, and we're here to archive them.
Thank you so much for listening, and special thanks to those who have helped us along the way. We hope you all have a great night.
Thank you very much.
Okay, that was a great talk. Thank you so much. Do we have some questions, people, that you want to answer? I know Kristen and Pam have questions, so go ahead, Kristen. Oh, that's clapping, sorry. This is why I shouldn't be the moderator. Okay, I can't see the symbols. But
I have a question.
Yes, good. Go for it, Sam.
So Benjamin, you mentioned that, you know, scientists could just go into the archives and find the right ones. Do you know what that would entail? Like, what would that look like? How would they, like, are they all labeled with like their best characteristics? Like, I'm sure they're not, but how would they go about finding the right one?
Yeah, so the scientists go and talk to the phages in the freezer and see which ones want help. No, I'm just joking. So the way that we do this is, and it was outlined, or like we sort of looked into it with Isabella's case, is that basically scientists can be like, okay, we need to get phages that destroy this specific type of bacteria. And so when they go to this archive, not only are these phages just being frozen in their samples, that way a sample could be sent to whoever needs to make the phage cocktail, but we also go through this process of making sure there's other information that's archived with them, such as what type of bacteria they infect and the type of life cycle that they undergo. So for like the phage therapy in Isabella's case, they wanted to look for phages that could infect Microbacterium abscessus, and were also lytic because that way they actually destroy the bacteria present. So scientists can just go and find that information and get sent phages that will hopefully have a good chance of infecting and destroying those bacteria.
Cool, thank you so much.
We have a question in the chat and there are 2 hands up. Dee Dee's asking, why is it useful to determine the cluster that a particular bacteriophage belongs to?
Basically, a cluster is grouping them together based off of genetic similarities. And since we don't know every single purpose of every single gene yet, what we do know is that like these phages have similar genes. And so the reason we value that is because it gives us a little bit more information on each individual phage, because we put them into groups based off of like homology, which allows us to kind of get a better understanding of one phage by comparing it to ones that we know for sure are going to be similar, because they must be similar if they have such a high homology in their genome sequences.
Okay. Brian, how about you?
Hello, can you hear me?
Yes, we can.
Okay, great. So during your presentation, you brought up various morphologies of these phages, such as non-contractile tails and tail spikes. I guess I have 2 questions, but I'll limit it to one. What function do those morphologies provide?
Yeah, so the sort of like idea behind the morphologies is again just sort of another classification system. When you have like, I don't know the exact number of how many phages exist in the world. I don't know if anyone truly knows that number, but phages are a very diverse sort of like virus type, and they come in all sorts of shapes and sizes. So beyond just those tailed Mycobacterium phages, there's like a whole host of them outside of that. So we just look at two specifically. And within these two, they can exhibit some slightly different properties. So I know that one of them, that's another tailed phage that we didn't talk about, is called Photovirus. It has this really short, stubby tail. They're kind of cute if you want to look into them. They have a unique way in which they actually can infect bacteria due to their short tail. Isn't actually long enough to go through the bacterial wall. So they have some extra proteins to help with that. So that's just one example of how the different morphotypes have some unique characteristics, but ultimately it's just another means of classification.
Okay,
So
and one more. Oh, sorry. Go ahead.
Sorry, so clarification. There is some functionality to it, to that tail morphology, for instance.
Yeah, so like with the Photovirus and their long tails, admittedly there is still research being done on them as phages are still pretty new, but it's hypothesized that the tail could help with the movement of the Photovirus, so they may be quicker than the Myroviridae. And the Myroviridae, they actually can like contract their tail during the infection, which could perhaps give some advantages. But this is again another area that we should probably research further into.
Thank you.